Welcome back again to a new video tutorial session from Biology Boostup. And in this video tutorial session, we going to study about the restriction endonuclease enzyme. So now let's start. The restriction enzyme, also called restriction endonuclease, a protein produced by bacteria that cleaves DNA at specific site along the molecule. In the bacterial cell, restriction enzymes cleave foreign DNA and thus eliminating infecting organisms. Restriction enzymes can be isolated from bacterial cells and used in laboratory to manipulate fragments of DNA such as those that contain genes. For this reason, they are indispensable tools of biotechnology or recombinant DNA technology. Next, the types of restriction endonuclease enzyme. At first, type 1 restriction endonuclease are complex endonuclease and have recognition sequence of about 15 base pair. They cleave about 1000 base pair away from the 5' end of the sequence TCA located within the recognition site. For example, ECOK, ECOB, etc. Next, the type 2 restriction endonuclease are mostly stable and induce cleavage either in most cases within or immediately outside their recognition sequences, which are symmetrical. More than 350 different types to endonuclease within over 100 different recognition sequences are known. They require Mg2+ ion for cleavage. The first type 2 enzyme to be isolated was HIND2 in 1970. Only type 2 restriction endonuclease are used for restriction mapping and gene cloning in view of their cleavage only at specific site. Now, the type 3 restriction endonuclease are intermediate between the type 1 and type 2 enzymes. They cleave DNA in the immediate vicinity of their recognition site. For example, ECOP1, ECOP15, HINF3, etc. Type 1 and type 3 restriction endonuclease are not used in gene cloning. The type 3 enzyme recognize asymmetric target site and cleave the DNA duplex on one side of the recognition sequence up to 20 base pair away. Now next, the naming of restriction endonuclease enzyme. The naming of restriction endonuclease enzyme is based on following rules. First, each enzyme name should have a 3-letter code. Second point is, the first letter of the code name is derived from the first title of the genus name of the bacteria from which it has been obtained. Third point is, the second and third letters are from the species name of the bacteria. Next, the 3-letter code name is followed by the strain number. If one strain has more than one restriction enzymes, those will be identified by Roman numbers as 1, 2, 3, etc. A general name endonuclease R may be added. For example, ECO R1 enzyme, which is derived from E. coli carrying antibiotic resistance plasmid RI. Next, the nature of cutting by restriction endonuclease enzyme. The restriction endonuclease enzyme cuts the DNA molecule by cleavage into two fashions. First one is blunt end fashion and second is streaky or cohesive end fashion. In case of blunt ends fashion, certain restriction enzymes like SMA1, HAE3 cleave the DNA duplex at the same point to produce blunt end fragments. The utility of this style during the joining of DNA fragments is that any pair of ends may join together irrespective of sequence. This is useful to join two different sequences without introducing any additional material between them. But in case of streaky or cohesive end fashion, most restriction enzymes like ECOR1, BMH1, HIND3, etc. makes single strand cut at some nucleotide pairs apart in the opposite strand of DNA and generate fragments having single-stranded tail or streaky end at the both sides of the end. Those fragments generated at a given restriction site are complementary to those on all other fragments generated by the same enzyme. At the room temperature, those single-stranded fragments can transiently base pair with those of other DNA fragments generated with same restriction enzyme, regardless of the source of the DNA. This is the streaky or cohesive end fashion cutting by restriction enzyme. Next, the important role of restriction endonuclease enzyme in biotechnology. The restriction enzymes are the basic tools of genetic engineering. Its importance in genetic engineering are: first, the use of different restriction enzymes allow the DNA of human genome or that of other organisms to be dissected into different set of specific fragments. Second, the number and size of the fragments generated by a restriction enzymes tells the frequency of occurrence of the target site in that DNA. Now, third point is, in order to join two fragments of DNA together, it is not essential that they are produced by the same restriction enzyme. Many different restriction enzymes produce compatible cohesive ends. For example, AGE1 and AVA1 produce molecules with identical 5' overhangs and so can be ligated together. Fourth point, methylation can reduces the susceptibility of DNA to cleavage by restriction endonuclease, and the efficiency of DNA transformation. Next, it is important to eliminate restriction systems in E. coli strain used as host for recombinant DNA. If the foreign DNA is introduced into an E. coli host, it may be attacked by restriction systems active in the host cell. Some strains like E. coli C and HB101 have deletion in restriction system and hence lack all restriction activities.